So let's move up to the next speaker. So that's another recorded presentation from Jonathan Holmes. Jonathan is a PhD student at the University of Leicester, and he studies the influence of hypermutable traction bacterial survival and immune evasion. So Lisa, if you could start the talk. Thank you.
Hello everyone. Hope you've all enjoyed the talk so far. My name is Jonathan Holmes. I'm from the Department of Genetics and Genome Biology at the University of Leicester. And my talk will focus on some of the work I've been doing on a relatively new bacterium, Campylobacter hepaticus, and how our understanding of its genetics and genomics might tell us a bit more about its role in spotty liver disease. So Campylobacter hepaticus was first isolated in 2015 during an investigation into spotty liver outbreaks on free-range poultry farms. And it was discovered while sampling the liver of the birds displaying spotty liver disease. And it was noted that there was a Campylobacter, parent Campylobacter, which had partial matches to C. jejuni and coli during PCR analysis. It was only after 16S sequencing that they confirmed the presence of a new Campylobacter hepaticus. So spotted liver disease is an acute infection that decreases egg yield and increases mortality rate in free-range layer chickens. Interestingly, spotted liver disease outbreaks rarely occur in battery farm chickens. It only really occurs in free-range poultry, which sort of leaves the door open to a possible transmission model from wild birds to domesticated birds. So spotted liver disease has been linked to Campylobacter hepaticus, but recently a infection model where chickens were infected with C. hepaticus, then later went on to show symptoms of spotted liver disease. So this kind of suggests that C. hepaticus plays a major role in causing spotted liver disease among poultry. So there's been a few genome analyses done already in C. hepaticus. In 2017, an analysis noted that C. hepaticus had a much smaller closed genome compared to its closest ancestor, C. jejuni, with a loss of about 20 kilobases. And this is put down to a reduction in iron acquisition and metabolism genes. But in addition to the loss of genes and loss of gene function, it was also noted that there were the presence of genes absent in C. jejuni, such as the detection of a cassette of genes involved in glucose metabolism, which are now used to identify C. hepaticus. And it was confirmed that these glucose metabolism genes were not only there, but they were also functional during utilization of glucose by C. hepaticus from the environment, which suggests that hepaticus can use alternative carbon sources to C. jejuni to help it sort of survival and growth. And so we wanted to then drill down on how hepaticus and what systems hepaticus can use to colonize the liver, because we know it as a liver colonizer, we know it can invade the liver, and we know it can go on to cause spotted liver disease. So what kind of alternative metabolism systems and other systems might be used by hepaticus to facilitate this invasion? So we selected a sample of 35 whole genome sequences from the UK and Australia. And we wanted to compare these to other Campylobacter species, so we selected an additional 15 reference strains. And then we selected 28 C. jejuni genomes, C. jejuni being hepaticus a close ancestor. And we wanted to sample these from the same sort of sources, so poultry, and from the same countries, the UK and Australia, for this direct comparison. So to begin with, we drilled down on what kind of gene loss and what kind of gene gain was going on here. So the dendrogram to the left compares our set of C. hepaticus isolates on the right to our C. jejuni isolates on the left, where black indicates the absence of a gene and white indicates the presence of a gene. So immediately, C. hepaticus contains a much smaller core genome and a very small accessory genome. There are very few, if any, genes that can be said to be differentially acquired between our C. hepaticus isolates, whereas in C. jejuni, across the same sort of sources and the same sort of time frames, you can see a quite wide range of genes being present in some isolates and absent in some isolates, sort of giving this quite wide accessory genome, which is absent in C. hepaticus. So then we can look at genes that are present in C. jejuni, present in C. hepaticus, but absent in the other. So we identified 74 genes in C. hepaticus that were not present in C. jejuni. And drilling down on these, we identified that the vast majority of them were in metabolism and transporter proteins. Transporter proteins group into two groups, which are those involved with sugar uptake and those involved with oligopeptide uptake. We also identified that there was very little variation between the UK and Australian clusters in terms of core genome size. So the metabolism genes presented quite an interesting line of inquiry. We identified the glucose metabolism cassettes that the previous genome investigations had detected. We also identified a apparently complete set of glycerol uptake and metabolism genes. So the schematic below shows the region of the hepaticus genome on top containing these glycerol uptake and metabolism genes, and the same loci and CGG on the bottom. So hepaticus strikingly has this GLPF, GLPK, and GLPD cassette of genes, which can allow for the uptake of glycerol and the breakdown of glycerol, and then the entry into the carbon metabolism system. So this might represent another carbon source that C. hepaticus can utilize in the environment additionally to glucose, which might help with its survival, say inside the liver. So we wanted to investigate how sort of base substitution rate differs between C. hepaticus and CG29. So we broke it down to our Australian and UK isolates. So the graph on the left, the scatter graphs on the left, show the percent identity between C. hepaticus and CG29, with the UK on the right and Australian to the left. So the distribution of our sort of base substitution rate was very similar between the UK and Australia, with about 85% identity, complete identity. And drilling down onto what was going on here, we noted that the vast majority of this divergence was due to sort of single polymorphisms, and very little was due to sort of the indels that might be cutting out large sectors of the genes, which indicated that, in fact, the genes that were present in G29 were present in hepaticus, and they probably were playing the same role in the cell. They were very unlikely to be orthologous genes. So this suggested that the core genome between G29 and hepaticus was not only conserved in its presence and absence, but also conserved in how it was made up, what kind of functions were going on, and what kind of systems they were playing a part of. So then we wanted to carry on and look at some other kind of survival and adaptation systems. One of the areas my lab works on is in phase variation. So we looked at the phasome of C. hepaticus, which is the combined number of phase variable genes that are present in that particular organism. So we immediately identified that C. hepaticus occupies a distinct niche from all the other Campylobacter species. So in the phylogenetic tree to the left, hepaticus is in gold and gray. All the other Campylobacter species are sort of branched off and intermingled, whereas hepaticus sort of occupies this quite distinct node of the tree. And while it occupies this node, the actual mechanism of phase variation remains pretty constant, with the vast majority of these hypervariable repeat tracks being poly G, which is conserved amongst all other Campylobacter species, with one minor exception. So while the mechanism of phase variation is conserved between C. hepaticus and other Campylobacter species, the genes that phase variation might be acting upon are very different. So we compared this to CG29, and we identified 3 clusters in C. hepaticus where phase variation was kind of concentrated. Well, we identified 2 clusters in CG29. These clusters represented quite sort of linked back quite nicely to C. hepaticus. So CG29 and C. hepaticus share about 2 clusters of phase variable genes. However, C. hepaticus maintains a 3rd cluster of genes which do not show any orthologous presence in CG29. So these phase variable genes are distinct in hepaticus. So drilling down on these, we noted that there are a few genes there which we could identify immediately as having functions involved in invasion, one of which was the 001265 gene, which codes for a transferase, which, while has no orthology to CG29, say TREB168, does have orthology to 81176, which is a highly infectious substrain of Campylobacter, which is detected in poultry, humans, and cattle, suggesting that hepaticus might have picked up this gene, and this might be possibly linking back to its invasion into the liver and the cause of spotted liver disease. So just to quickly summarize what I've been talking about, so we've outlined some of the context of the data surrounding C. hepaticus, and we've sort of linked in what we've done in context of this research. So we've identified a few possible recombination selection events that occurred across a range of novel genes. So it suggested the metabolism systems that C. hepaticus can utilize for occupation. And we sort of outlined the role of phase variation in C. hepaticus invasion and how this might play a role in spotty liver disease. So that just leaves me to thank you for listening. I'm Kurt. This is a pre-recorded talk, but please, if you have any questions, feel free to contact me by my email address. I'd be happy to take any questions from there. Thank you very much.
So thank you, Jonathan. Actually, he's not in the chat, so if you have any questions, please email them directly to him, as he said, or put them on Discord and have a ongoing discussion